Hello and welcome to Principle of Cognitive Neuroscience Part A. As you may know, the course is divided in three parts. Main models, where we explain how the brain functions, how people think the brain functions. Main concept that constitutes the fundamental element that represent a result in neuroscience, and practical sessions that will not be recorded, where I will teach you how to practically do science and use softwares and write papers and get funding. Most of what I'm going to describe in this lecture is written in this paper, Beyond Cortical Localization in Clinical Anatomical Correlations, and in my book, Atlas of Human-Brain Connections. So, associationism. Associationism is a model of the functioning of the human brain, which is based on the association between brain regions. And this is only possible if you change the way you imagine or see the brain. In the previous description that I gave to you, the localizationism, people mostly explore the brain looking at its surface, as you can see on the left, looking at the different gyri and sulci, eventually cut the brain into slices to discover subcortical structures such as the thalamus or the striatum. Cannot show you. But all these areas in the brain and all these structures in the brain are interconnected thanks to white matter connections that you can see on the right here. So you have a different kind of connection. You have the one that connects the two hemispheres, the one that connects subcortical structures such as the thalamus and striatum with the cortex, and you have the one that link together brain areas. And these connections are not a recent finding. The description of white matter dates the 16th century with like a postmortem dissection. As you see, at that time it was quite intense. We were not even like severing the head from the body, and like the head was like just sliced on the body itself to do anatomical investigations. But really thanks to Niccolò Steno in the 17th century that we started like really being curious about white matter and what white matter connections can do in the brain. And you can see it in his writing when he's saying that fibers are disposed in the most art-filled manner, since all the diversities of our sensations depend upon them. We admire the contrivance of the fibers of every muscle, and ought still more to admire their disposition in the brain, where an infinite number of them contained in a very small space, each executes their particular offices without confusion or disorder. And because of this specific order that they have, anatomy started trying to map them and show their organization. And so you see this first drawing from Raymond Vuissens in the 18th century showing the organization of the projection systems. This is the first depiction of the corticospinal tract. But looking at the anatomy itself was not enough to really make us understand how it would work. And it really took a true genius to understand, have the intuition on how the system of connection of the brain work. And it's really the work of Isaac Newton, like just his intuition, what he wrote at the end of his book, that gave the spark onto investigating and understanding the extraordinary role of white matter connection in the brain. And so we'll read it together. This is the last page of his mathematical principle, where he wrote that, and now we might add something concerning a certain most subtle spirit which pervades and lies hid in all gross body, by the force and action of which spirit the particles of bodies mutually attract one another at near distances and cohere if contiguous, and electric bodies operates to greater distances, as well repelling and attracting the neighboring corpuscles. And light is emitted, reflected, refracted, inflected, and hits bodies, and all sensation is excited, and the member of the animal bodies move at the command of the will, namely by the vibrations of the spirit, mutually propagated along the solid filaments of the nerves, from the outward organs of sense to the brain, and from the brain into the muscles. But these are things that cannot be explained in few words, nor are we furnished with that sufficiently of experiments which is required to an accurate determination and demonstration of the laws by which this electric and elastic spirit operates. A true genius, just, after defining the law of relativity, put down like as the essential functioning of the other mystery, which is the human brain. And now that we had a model and an idea really of the fibers conveying information and helping to produce behavior through the exchange between the brain and the body, this motivated even more anatomies that carried on their work drawing and defining the anatomy of these main connections. You can see the work of Johann Christian Rell on the left of these slides in A and B, whereby you see the first depiction of the Arcuate fasciculus, main tract for language, curiously drawn for the first time in the right hemisphere, and the ancinate and inferior longitudinal fasciculi, which are also other tracts connecting respectively frontal and temporal and occipital and temporal lobes. And Christian Rell work was pursued by a student of Carl Friedrich Burdach, who was able to describe main white matter connections such as the cingulum or as a projection system. And this was nice, and we needed to group them together to structure them under the same head. And Theodor Meinert, 19th century, came out with this classification that is still of use today of connections that are linking subcortical structure as well as structure of the spinal cord with the cortex. And because these fibers are going from the inside of the brain to the outside of the brain, he called them projection fibers. Then another group of fibers link the left hemisphere with the right hemisphere and the right hemisphere with the left hemisphere. And because they link these two hemispheres, Theodor Meinert called it commissural fibers. And finally, a third group of fibers connect regions within the same hemisphere, allowing for the interaction between brain regions within the same hemisphere through a mechanism that build an entire school, that is like the basis of an entire school of thought, which is the associationism, and it's coming from the existence of association fibers. This connection associates the function between different areas in the brain. And so Theodor Mine had like draw some hypothesis on how should it work, and he has this example of a child representing essentially a human being not knowing anything about the outside world, and children are attracted by what is shiny, sorry. And you can see how this child is looking at the candle with his eye, and his eye being further connected to the occipital lobe, labeled here OCC. And because it's shiny, he's going to try to grab it with his hand, as you can see here. And the hand is connected to the hand is connected to the sensory centers over here. As you can see, like the little arrows indicating that information is going along the blue line, which represents a connection. And this information is going to be conveyed is the pain of this child burning his hand on the flame. And the brain is going to make an association between the pain and the fact that you should remove your hand, which will be the red fibers going towards the hand with the little arrows going towards the hand, indicating to the hand to retract. And so you see how you have a series of events happening. The vision of the flame, that is associating with like the motricity of the hand. They're going to try to catch a flame, the sensation of burning, and the removing of the hand. You have a serial organization of events that is learned by the brain, so that the child, when he want to try to reach the flame, will remember this sequence of events and will not burn himself, because he will not try to touch a flame anymore. And this is like the fundamental idea between the associationism. The brain areas associate their activity in order to optimize our behavior and increase our chance of survival. So like other way of exploring anatomy, as I said, we have a nomenclature when we study white matter connections. So you see, for example, for traditional surface anatomy, on the top left, you have, when you're like in the towards the front of the brain, it's called the rostral part of the brain. When you are towards the posterior part, you're going to call it the caudal part of the brain. When you toward the top, you like the dorsal part, and then toward like the inferior part is going to be called the ventral part of the brain. And if you slice the brain, you will have different axes, and usually we call them axial if you slice your brain from the front to the back, coronal if you slice your brain from the top to the bottom, and sagittal if you slice it sideways from one ear to another. And we can build coordinate along these three axes to locate ourselves. In the domain of white matter connections, we're using three main classifications, as I said just before. Projection fibers going from a subcortical structure to the surface of the brain, or from the spinal cord to the surface of the brain. Association fibers linking together brain areas, and commissural linking together brain areas in the same hemisphere, and commissural fibers linking the two hemispheres together. Now, it's interesting, but why does it matter really to know about the connections in the brain? Are they really important? If we believe like the theory of Constantin von Monaco, who was the first to push forward the principle of diaschisis, yes, they are really important. In these drawings that he made at the beginning of the 20th century, you can see the lesion called Erd, it's German, sorry about that, in the right hemisphere, and you can see how the lesion damage the surface of the brain and the neurons and interneurons that you have here. But as the lesion is going further deep inside the brain, it also disconnects, not only connection stopping in the region, but also connection passing by, but not stopping in the region that has been damaged. So you can see here like the deconnection of green connections that are connecting within the same hemisphere brain areas. Those are associative connections. You have also the blue connections that are projections and commissural connections. And you have those red connections that are like, you know, because we cannot go through the brain, but they're like passing by and going to a different place in the brain. And those have been disconnected as well. And so the idea is because you disconnect those axons, you will, like the lesion will have an impact, a distant impact on the brain, on the projection of those axons. And this is essentially what is behind the concept of diaschisis. You can either, through the mechanism of disconnection, inhibit an area that used to be excited, or disinhibit an area that used to be inhibited. And this will have obviously an impact onto the behavior, the functioning of the brain, and thus you will observe symptoms associated to that. Now you might think, well, okay, this is an interesting concept, but how it relates to what we're doing today? Well, it relates quite a lot, because I described to you in the previous lecture localizationism, which is a concept that has been invented by Joseph Franz Gall, who was doing the phrenology, touching the skull, and imagining that like the shape of the brain behind it is gonna deform the skull, and you can touch bumps and this reflect like the shape of the brain behind it. And the idea was like, if one brain region is bigger, you'll be better at the function that this brain region is doing. Really, like, you know, a concept that we still use today with voxel-based morphometry, for example, VBM is what it's exactly the same thing. Except that we look at MRI and directly the brain and not at the skull. And this concept has really been championed by Paul Broca, who showed that if you have a lesion in one brain area, typically the Broca area, posterior part of the inferior frontal gyrus, you will have always a production aphasia or Broca aphasia, because you have here a center that is essential for the production of language. According to the theory, as I said before, the brain is fractionated like a mosaic of brain regions dedicated to specific function. And the damage to a single brain area is just gonna have an impact on this area and no neighboring areas at all, because it's all, you know, isolated. This concept is directly assessed through neuroimaging methods such as voxel-based morphometry, but also when you're doing classical voxel lesion symptom mapping, you assess voxel in isolation cortical thickness is the same, or task-related fMRI, PET EEG, MEG. If you do not capture the interaction between brain areas in your model, you are in the localizationism school. On the other hand, you have another school of thought, which is as old as the localizationism, that have been set up by Theodore Minot with the discovery and the classification of association fibers. He pushed forward the idea that brain areas can associate their activity so that they can produce behavior or optimize behavior. And this has been championed by Carl Wernicke with a model of conduction aphasia, where the production of language can be disconnected from the comprehension of language and lead to a deficit which is called conduction aphasia, whereby if I tell you tomorrow I have to go to the store to buy some Coca-Cola or Coke, you will, and I ask you to repeat what I said, you'll say that I said I was going to buy some drinks at the supermarket, not today, but the day after. And so like the concept is still there, but the exact words are not the same, just because the link between the comprehension and the production has been severed. According to this model, every regions are working in interaction with other regions through a network, and damage to one brain areas will have an impact to the areas it is connected with. And several methods can capture this relationship. You have track-wise analysis, which I build mostly, functional connectivity, effective connectivity, and anatomical connectivity. Track-wise analysis are using models of how the brain is connected to help understand the interaction between and modeled interaction between brain areas. Functional connectivity is looking at the correlation, the synchronization of activity between brain areas. We'll come to that point during the holistic lecture. Effective connectivity will measure whether the activity of one area has a significant impact on another area. And anatomical connectivity will directly measure the wires that are supporting these connections. I'll focus mostly for this lecture on anatomical connectivity and track-wise analysis. I'll keep functional connectivity, effective connectivity for the holistic approach. So anatomical connectivity can be derived from diffusion weighted imaging tractography. Diffusion weighted imaging has been first reported in 1994 by Peter Basser and Denis Le Bihan, who invented this sequence in the MRI scan. And the first thing that they measured was a piece of beefsteak that they put in the MRI scan. And what they wanted to look at is diffusion of water molecule. And as the diffusion of water molecule might be constrained by the structure of filament into a tissue, they thought they could estimate indirectly, via measuring the movement of water molecules, the structure of the tissues they put in the MRI scan, which they did here. You can see this beautiful, gorgeous 1994 mathematical models. On the left, you have the first acquisition of this piece of beefsteak in the MRI scan, where you can see that the sphere is going left to right. And then Denis Le Bihan asked Peter Basser to get in the MRI scan and turn a little bit the piece of steak, and you can see that the model of orientation of the fibers inside this tissue has been changing, and the ellipse have been rotating a little bit, indicating for the first time that it was possible to quantify the orientation of tissue fibers of the muscle of the steak by using diffusion weighted imaging. And of course, in the papers, it says that a potential application for this wonderful method might be the brain. Diffusion MRI is, as I said, working on the measurement of the diffusion of water molecule. And as you can see on the top left, that's a magnified voxel in the cortex, where the neurons are distributed quite aleatorily, and there is no structural organization that will constrain the diffusion of water molecule there. What the MRI scan is going to do, it's going to record signals that we can model with the model of the tensor, and the tensor will represent a little bit the diffusion, the average diffusion of water that look like a sphere here, because there is no preferential direction of the diffusion of water inside this tissue. On the right panel, you have a high resolution voxel slice of white matter connections. And you will see that water is constrained. It diffuses essentially inside the axons, but not across. And the MRI scan can record a signal that is summarized thanks to the tensor model as an ellipse, which is elongated, deformed really according to the shape, according to the main direction of the diffusion. So you have these long cigars, and you get to use that water molecule are going in that direction in this precise voxel. Then the idea was to piece together voxels together, to build trajectory in 2 dimensions, and to do it inside the brain in B, and then to move in 3 dimensions in C to obtain those tubes that happen to be white matter fasciculi. Okay. So at first we were like, is it like really true anatomy or just a fancy mathematical reconstruction? And if you look at typical white matter tracts, such as the Arcuate fasciculus, you can see that there is a match between standard post-mortem dissection of white matter connections and what we can obtain with diffusion weighted imaging on the right, except that on the left it was post-mortem, on the right it is in the living human brain, and you can do as many people as you want. That's great. And so the first thing people started to do is to study anatomy and revisit the anatomy of white matter connections. You have an example here whereby we can see that the Arcuate fasciculus that we traditionally describe as a connection between the Broca area and the Wernicke area is not as simple as we thought it was. So you do have this direct connection between the Broca and the Wernicke, but you also have extra connections that make a stop inside the parietal lobe. So you have a long segment, the traditional Arcuate fasciculus, and then you have 2 short adjacent segments of white matter connections that are going from Broca to the parietal lobe, Geschwin territory, mostly supramarginal gyrus, and a posterior segment that is starting from the same area and then projecting onto the Wernicke territory. Okay. Now, you know, this is mathematical reconstructions. How true is that? Well, you see on the right part of this slide that if you compare with postmortem dissection, then tractography can help us to guide our postmortem dissection and put in evidence demonstrate the existence, this time in postmortem, of something that we found in tractography, but this time in postmortem dissections. Because we can scan so many people, it is possible to also assess how different people are one from the other. And if you look at the system of the long arcuate fasciculus connecting Broca and Wernicke territory, and these two short segments, the green and the yellow, you can look at how they are lateralized between the left and the right hemisphere. Given that this connection should be dedicated to language, we expect a strong left lateralization. Is that true? Yes, you can find a strong lateralization for the standard arcuates that connect Broca to Wernicke in about 60 to 80% of the population. They are going to show an extreme to a medium lateralization of the arcuate fasciculus. But some people, about 20% of the population, will not show any lateralization at all, and they will have a very symmetrical arcuate fasciculus. And this has an impact onto how they'll be able to remember a list of groceries at the supermarket, at the California Verbal Learning Test that you can see here in the C panel. And there are also differences between male and females. Males tend to be slightly more lateralized than females, although as you can see, you have 40% of females that are like having a strong lateralization, and then they're more like distributed among the three groups, while male are like mostly strongly left lateralized. Be like, okay, but white matter variability is interesting, but I don't see why should it matter at all? Like, why would we be interested into that? It's just fundamental science. Well, excellent question. The reason why we should be interested about that is because sometimes you can injure your brain. Now, just let's do a little mental experiment. Just imagine that you have a stroke in your left hemisphere, and you damage this set of connections that we see here in the left hemisphere. Would you rather be in the group 1 and have a strong left lateralization? Or would you rather be in the group 2 and have a small lateralization, or the group 3 having a symmetrical organization of your arcuate fasciculus? I'm pretty sure you'd like to be in group 3 because, at least you have a backup in the right hemisphere. And this is a work of my colleague Stephanie Fockel, who's been able to demonstrate that after a stroke in the left hemisphere, the size of your long segment, the typical arcuate fasciculus, the right branch in the right hemisphere, actually do matter a lot when it comes to outcome in the language. So if you take the example here of 3 patients, patient 1, 59 years old, very small arcuate in the right hemisphere, got a stroke in the left hemisphere and came out of his stroke with an abnormal language as defined by the aphasia quotient. You can see here in the scatter plot. Patient 2, despite being older than patient 1, had a stronger arcuate fasciculus in the right hemisphere, and 6 months after a stroke, showed still some difficulties to have to express herself, and the language was not judged really good, but better than patient 1. And then patient 3 that has a very strong arcuate fasciculus in the right hemisphere. 6 months after his stroke, was able to show a normal language despite being older than patient 1 and patient 2, which, you know, age is usually not a very good prognostic for recovery. You can also ask yourself questions about the ontogenesis, the development of the arcuate fasciculus. You know, are we born with a lateralized arcuate fasciculus, or did it become lateralized along our life or our brain maturation? We did explore that, and we were not able to evidence any differences in the lateralization of the arcuate fasciculus from the age of 10 to the age of 40. So it probably happened before that. Unfortunately, we couldn't scan people before 10 years old, but apparently, yeah, it happened before that. Another interesting thing is to explore the phylogenesis, and you can do that through the investigation of comparative anatomy. You can look at the brain of other species, usually primates, because it needs to be close enough to our brains, and use this information to really understand what is making us so special, humans, making us humans so special. And also, if you look at the arcuate fasciculus in macaque monkeys, it actually doesn't look like an arcuate fasciculus, and it is very small in comparison to the human. So mostly, you know, the monkey arcuate fasciculus will look like the little red segments that we have over here, and then you see how it expanded like drastically in humans. For the blue part is like our arcuate fasciculus is way bigger in the left hemisphere than it is in monkey in proportion. I mean, and that is because humans like us have been able to elaborate such a complex and refined way of communicating between each other through words, whether they are exchanged through an auditory verbal channel, or writing words when they exchange through a motor or visual channel when you're writing something. Speaking of writing words, we also been able to explore and investigate whether white matter connections can be different between people who are literates and learn to read at school, and people that are illiterate that never learn to read. Believe me, it's not easy to find illiterate people nowadays. Those who are like in eccentric countries of Brazil, very difficult to build a group like that. And we also had the chance to have an extra group of participants who used to be illiterate, but learned to read late in life. So we compared those three connections in these three groups, and we found like a strong relationship between how water molecule diffuse inside the yellow connection. You remember connecting the Wernicke area with inferior parietal lobule. And we found like a strong relationship between how water molecule diffuse inside that structure and your performance at a reading score. How many words were you able to read in 2 minutes? And this was very significant, and it was also different if you do just a group comparison between illiterate and literate people, it was also different between illiterate and ex-illiterate people, but it was not different anymore between ex-illiterate and literate people. That means that through the acquisition of literacy, the brain might change is a yellow connection of the arcuate fasciculus, connections between Wernicke and supramarginal gyrus and the angular gyrus, and this change actually might even happen late in life when you're 40 years old. And so that was like really exciting, and again, but the hypothesis that was behind it is because through this connection you can make an association between the written representation of the word and its auditory semantic representation. And then for the lower part of the chart, we also checked that it was significantly different at any portion of the tracts, which you can see it was. Then, you know, again, you can tell me, well, this is very interesting, but, you know, this is just LC controlled. We can see that you can change your brain and this is this connection. Well, but how does it matter? Why is it so important? Well, it is important because if you learn how different portions of the white matter connection contribute to function, maybe you can help neurosurgeon not to do any permanent damage to the brain. Let me give you an example here, which is typical of a patient coming with an epilepsy. Again, if you've seen the other lecture, it never end well. So this patient is coming with an epilepsy, intractable, like drugs do not work. So he speaks with a neurosurgeon. So neurosurgeon recommend that we do surgery and remove the area of the brain that is supposed to start what we call the epileptogen focus. That's the area that lead to the epileptic crisis. And because this area is located in a brain area, in an area that is very close to the area we call the visual word from area that we spoke about in the last lecture, the neurosurgeon think, well, you know, we're going to remove the cortex, but we're going to be very careful not touching the visual word from area because we do not want these patients to come out of the surgery with an alexia, which would be the inability to read. So in order to avoid that, the neurosurgeon is asking scientists to put the patient in an MRI scan and show him like written words and control condition, do a contrast between the two, and get a P005 significant activation in the brain that is related to the visual word form area. And this is what you see on the left. This is a beautiful activation of the visual word form area. And then the neurosurgeon said, like, fine, I know where it is now. So what I'm going to do, I'm just going to cut the brain behind the visual word form area in order to avoid inducing any permanent damage. And that's what it does. You see like this nice triangle in the cortex that correspond to the damage, the surgery made to remove the epileptogen focus. Unfortunately, the patients come out of the surgery with a pure alexia, inability to read, not a single word. So what do you think happened? Yeah. well, I'm going to tell you, it doesn't matter that you don't touch the visual word form area if you cut the connections that are providing information to the visual word form area. If you cut the connection that will link the visual cortex with the visual word form area, you won't be able to read. If you cut the connections that link the visual word form area from the auditory semantic context, you won't be able to read either. And that's what happened. So later on, like the researchers were able to do some tractography, because they had some tractography before the surgery. They've been able to compare the tractography before and after the surgery and see that actually the neurosurgeon in that case deafferented the visual word form area from any visual input. And that's why the patient was showing pure alexia. And that's also why white matter connections are so important to know. The way white matter connections can help understand how you can do surgery has later on been pushed forward by Hugues Dufaux, who's been able to demonstrate that the typical way is going to do a temporal surgery is limited by the subcortical anatomy. If you were to do clinical neuroanatomical correlation with neurosurgeon and your model is a temporal lobe surgical resection, it's going to be really boring because they do always the same lesion. There's very small variability there. And this is mostly because they are limited in the way they can do the surgery by several factors. They cannot go too far back because the arcuate fasciculus is going to start being responsive and the patient will not be able to speak. They cannot go too deep because there are the optic radiation and then the patient won't be able to see anymore. And that's why they just do this typical cut that you have here. And you know, tractography is great and all, and you can actually extract numerous beautiful connections that looks like the real deal on because this is a teaching lecture. I will go through them, and so you can like start learning their anatomy and their name. On top left, you have the arcuate fasciculus. Middle top, you have the anterior commissure. On the top right, you have the uncinate fasciculus. On the middle left, you have the inferior fronto-occipital fasciculus. Then you have the inferior longitudinal fasciculus. Then you have the arcuate fasciculus. On the right middle, you have the cingulum. Bottom left, you have the fornix. Then middle bottom, you have projection, the projection system, internal capsule. And then on the bottom right, you have the cerebellar connections, including corticopontocerebellar, spinocerebellar, and here you have the lateral vision of the same connections. I engage you to try to challenge yourself and giving the name. I'll give it to you quite fast for the purpose of not being too boring, but it's good you learned that. So you have the corpus callosum, anterior commissure, uncinate, inferior fronto-occipital fasciculus connecting the frontal lobe with the occipital lobe, inferior longitudinal fasciculus connecting the occipital lobe with the temporal lobe, arcuate fasciculus, cingulum, fornix, internal capsule, and cerebellar pathways. So they look great and all, but are they accurate enough to guide surgery? Well, while a lot of people would say yes, I've been in the field long enough to tell you that you should be careful. And typically, if you look, for example, at the corpus callosum, you can see it's a beautiful reconstruction in humans using tractography, deterministic tractography. You have beautiful corpus callosum that project to the middle section here on the right. But if you look at postmortem dissections on the left, top left, notation is really showing that this corpus callosum should project not only on medial structure, but also on lateral structures. And at the bottom left, this is axonal tracing in monkeys, which should be our gold standard. And you can see that the surface of the cortex of the monkey is not uniformly connected by commissural corpus callosum fibers. You have like some holes in there, but the lateral portion is enough connected so that we would expect to have lateral projection of the tractography. And it is not the case here. And that's a problem of tractography itself. And then if you learn more about tractography yourself, some people will tell you, well, you can use probabilistic tractography to get these fibers, because if you lower the probability, you will get lateral projections. And while it's partially true that you get this lateral projection, you get them at a great expense of connections that are not the corpus callosum at all. And you can see it here as we're lowering, we start having connections that have nothing to do with the corpus callosum at all. And this is a problem of probabilistic tractography as well. So, while tractography, standard tractography allow us to show really the core of some main white matter track, it is not good enough to depict exactly the anatomy of those connections like axonal tracing or wet science can do. But you'll tell me like, yeah, this is interesting, but who cares about the corpus callosum? It doesn't do anything anyway. That's fine. Let's speak about something that matters and speak about like the corticospinal tract, for example, where on the left you have postmortem reconstruction of the corticospinal tract that have been done by Rademacher and et al. That shows beautifully how the corticospinal tract should look like. And on the right, you have my atlas based on standard tractography that show you what tractography is showing you for the corticospinal tract. And you can see that they are quite different. I mean, like we're missing the lateral projections. Again, you might argue that if you were to use probabilistic tractography, you will recover those lateral projections. I tried it up, and yes, you can recover those lateral projections, but again, at the expenses of having so many extra connections that are not part of the corticospinal tract. So yes, it is a problem, and it does matter, because then you hate to see some papers like this, where people report that fiber tracking does not accurately estimate size of fiber bundle in pathological condition. This is the surgery of a low-grade glioma in a kid, where they remove the low-grade glioma and they cut the corticospinal tract. Yes, great. No more glioma, but the poor kid is hemiplegic now. And that's a prime of not having accurate methods that are accurate enough to really assess anatomy at the individual level. Fortunately, we're making some progress, and we can now reconstruct those connections that project on the lateral surface of the cortex for for the corpus callosum, as you can see here. On the left, you have your corpus callosum that is restricted by the internal capsule or corona radiata. And you can see it cannot go through. That's the standard tractography. And on the right, you have advanced tractography, typically spherical deconvolution, where you can see that those white matter tracts peer through the internal capsule and project on the lateral portion of the cortex. And the same goes for the corticospinal tract. We can now have its lateral projection to some extent, and we revised our atlas by providing all these tracts now with this advanced model, including all the crossing fibers. Having an atlas is convenient because it allows you to do what we call the tract-wise analysis, which are like you already have a knowledge of how the brain is connected, and you use that to interpret the result from the lesion of a patient. And typically something that we develop with our software, the PCB toolkit that you can download on our website, pcblab.com, was to use a patient, bring it into a stereotaxic space that we call the MNI space. We call that normalizing the patient. Overlapping a white matter tract onto this patient brain, because now they're in the same space, you know, the atlas of the white matter tract and the lesion of the patient are both in the MNI space, in the stereotaxic space, so we can overlap it, and then derive from that a probability of disconnection. This probability of disconnection is just an Excel spreadsheet, so you can run it into any statistical software and try to investigate what is the relationship between the probability of being disconnected and the probability of showing any disorder. And that's what I did for visual neglect. So for those of you who don't know about visual neglect, this is an example. So we ask the patient to cancel every line on this piece of paper, and you'll see that he's only canceling the line on the right side of the paper, but not on the left side, and that's not because of...
Vous avez arrêté de trait de la feuille?
It's not because of...
Ça, c'est bien. À peu près. Il en manque pas. C'est bon, vous les avez tous là?
Je sais pas si je les ai tous.
Essayez de chercher un peu.
It's not a visual impairment, it's a problem of like access to his awareness for the left part of the paper.
C'est bon, hein, vous me dites quand vous les avez tous. Ouais, tout le temps.
Je sais pas si je les ai tous. Hein.
C'est bon?
J'espère que c'est bon.
Okay.
So, as you can see, the patient only explores the right part of the piece of paper, never cancels the part on the left. This is a syndrome that is called hemispatial neglect, and that happens in 80% of the case after a stroke in the right hemisphere. Some of these patients recover, some others do not, about half of them recover. But for the one who do not recover, it would be good to know what was their lesion and how this relates to their recovery or not. And so that's what we did. We explored like the typical disconnection in this patient, and we found that out of all the disconnections that were possible to investigate, the one that was the most predictive of visual neglect was a disconnection of the frontoparietal segment of the arcuate fasciculus. It's like the superior nostril fasciculus that I'll come back to in a minute. And just this, if you look at the lesion and you look at its overlapping with the frontoparietal connection, this allows you to predict the fact that the patient will show visual neglect that is persistent after a stroke in 4 out of 5 cases, and also allow you to cancel out, like to demonstrate that, to put aside 19 out of 20 patients that do not have this disconnection and will not show a persistent visual neglect after their stroke. So it's a very strong predictor. Now we spoke about these 3 guys in the previous lecture, and so we had Phineas Gage, which is a typical case of a patient that had a lesion in the ventral part of his frontal lobe that led him to have a radical change in his personality and deficit in planning, going outside with thin boots and no jacket when it's raining. The case of Le Bon, who had a lesion of the inferior, posterior part of the inferior frontal lobe, and not able anymore to produce language, typically an aphasia, a Broca aphasia, production aphasia. And Louis and Henri Molaison, who had a surgery, bilateral surgery of his medial temporal lobe, disconnecting, bilateral lesion of his medial temporal lobe, damaging the hippocampus bilaterally, and leading him to have a severe deficit in the in memory, in the creation of new episodic memory. So what, like what's interesting is like those 3 cases are always seen as like really the 3 pillars of neuroscience, being medial frontal lobe, personality and behavior, posterior part of the inferior frontal gyrus, language production, hippocampus, episodic memory, declarative memory. And so what happens if you just try to do the same thing and look at the disconnection in this patient? Does it change the vision and explain better what is going on? And yes, it does. In fact, if you look at Phineas Gage, the bar interrupted the passage of main connections such as the uncinate fasciculus and frontal connections. And this might explain, because in cats, if you cut the uncinate fasciculus, they start developing a very aggressive behavior. The uncinate fasciculus is in yellow on the left. They can start having a very aggressive behavior, and that's because the frontal lobe doesn't have the possibility to inhibit anymore the amygdala, where you have all this emotional outburst. And then the bar also disconnected a lot of frontal connections that are essential for decision making and planning of the behavior. And by disconnecting that, this might explain also why Phineas Gage starting to change radically his behavior after his damage. In the case of Louis-Victor Le Bon, we can see that the lesion disconnected strongly all the perisylvian language network, including the arcuate fasciculus that we talk about, but also frontoparietal connection and the frontal ascending tract. So later on, we even showed that the frontal ascending tract is related a lot to the ability of to fluencies. And now, like when you look at that, you're like, oh, maybe it's not really the lesion in the brain areas that that led to that, but maybe it's a disconnection of this entire system that led to the symptom in this patient. Finally, you get the case of Henry Molaison that had a disconnection, bilateral disconnection of a system which we call the limbic system, essentially. So the bilateral disconnection disconnected phonics, which is really important for the acquisition of episodic memory. We know it because if you go in, if you see patients that have a cyst in the third colloid and they have surgery of this cyst, because the cyst is very close to the fornix. Sometimes the neurosurgeon damage the fornix, and the patient come out with very strong memory deficits. Yeah, like Henri Molaison also had a disconnection of the uncinate, which in monkeys have been associated to the building of emotional memories. You might know that a memory and emotion are very closely related, and a good way to remember something is to associate it with an emotion. So if it's disconnected bilaterally, we can imagine that this will have an impact on the memory of Henri Molaison. And then finally disconnected also the cingulum tract in blue. The cingulum tract connects the hippocampus, the medial temporal cortex, with the retrosplenial cortex. It's in the medial part behind the corpus callosum. This area is very important because it's been reported as being the area that lose most of its cortical thickness in Alzheimer's disease, shows most of the amyloid plaque and most of hypometabolism in Alzheimer's disease. Alzheimer's disease being a syndrome that is extremely related to the loss of memory. Now, if you know, like the dream is like if you repeat it enough in enough patients with behavior, you can build this map of the function of white matter connections. And we had the chance this year actually to finally produce the first map that is like showing like all the white matter connections here and their association with different functions. And you can see here how this is, you know, this is a multidimensional map, and here you can only see like really the top contribution of some function, but you can see how the left and the right hemisphere are dedicated to quite a different functions, and they have you have like different function happening through the association between brain areas. I'll come back to this during the epistemology course. So, you know, what would be the take-home message of this is that like we've been studying connections, but connections are infinitely more complex than studying neuronal soma. And you know, like, so when you study the entire connection of the brain, we speak about the connectome. And so although we've been able to decipher the genome, I don't think we'll be able to decipher the connectome, because the true connectome is at the microscopic level. And there is one person been working on it on mice for the past 20 years. It's Sebastian Seung. He wrote this fantastic book called The Connectome, How the Brain Wiring Makes Us Who We Are. It is a great book. But just in mice, it takes so much space, so much data. It's incredible. On the other hand, you have Olaf Spahn, who work on the connectome in humans, but this is derived from neuroimaging, and this is just, you know, it's like really the gross connectome of how brain regions speak together. It is not as refined as the true way we are connected. And the truth is, even though we were spending millions of euros, billions of euros to depict the connectome in a single human being, this connectome change, and your connectome before you listen to my lecture and your connectome after you listen to my lecture, at least I hope, already changed a little bit. And some, you know, like to give you a little representation of how vast is the connectome, you know, we have as many neurons in the brain as there are not stars that are visible with the naked eye in the sky. But we have 1,000 more connection per neuron. So each neuron is connected to 1,000 other neurons in average. That gives you the incommensurable level of connections there will be to map if we wanted to do a full depiction of the connectome, full and complete and comprehensive depiction of the connectome. So I'm afraid the best is yet to come. I'll leave you with a new method that is promising for us to explore the connection in the brain, and I hope you have the chance to learn from it and maybe use it one day to depict the full connectome of a human brain.
We're heading into the center of a mouse's brain, into the hippocampus, where memories are formed. Looking up, you can see neurons projecting to the surface of the brain. This is the work of Carl Diceroth and his team at Stanford University. By making the entire brain transparent, they were able to image it using a light microscope. They call the new technique clarity. Existing techniques for studying the brain's wiring are often limited to looking at very small volumes of brain, or they don't allow you to label genes or chemicals of interest. The advantage of clarity is that you can label lots of molecules in whole brains. So how do you make a brain transparent? The thing that obscures the view is fat. Lipid layers surround each cell. To remove them without disrupting the cell structure, the team used a hydrogel to create a mesh to hold the rest of the components in place. Then they could clear away the fat. This is a mouse brain before and after. The brain is now transparent to light, but it's also permeable to molecules, which means scientists can add molecular markers to highlight specific features. In this 1 millimeter block of hippocampus, excitatory neurons are green, inhibitory neurons are red, and cells called astrocytes are blue. The technique works in human brains too. This is a chunk of the frontal lobe of a 7-year-old boy who had autism. It's possible to trace the path of a single nerve projection through a forest of other cells. When the team looked closely in one layer of the cortex, they noticed ladder-like patterns where neurons had connected back to themselves and to other neurons. Similar abnormal structures have been seen in animals with autism-like behaviours. Being able to analyse brain structures like this and match them up with molecular information could help neuroscientists uncover how changes to the brain underlie disease. And it's not just the brain. The researchers say the technique could be applied to any organ.
So cool. All right, so I'll leave you here with the associationism. We'll see in the next course that it's not only about serial communication in the brain, it might also be about a true interaction between brain areas where the whole of their activity is more than the sum of their different parts. I invite you to join us the 11th and the 13th of November for the Brain Structure and Function Special Issue Symposium that is organized by the Clinical Neuroanatomy Seminars on the structural connectivity of the cerebral cortex. You can watch it on YouTube replay of the Clinical Neuroanatomical Seminars. I also invite you to join us for the Neurochino every Monday morning from 9:30 to 10:30 a.m. Paris time, where we speak about last week's science, what is exciting about what happened last week. Grab your coffee and come discuss with us. We'll be delighted to have you. In terms of grade and evaluation, I'll just remind you that the way you'll be graded for for this module is a mix between your attendance to the practical lecture, your participation in class, which this year, due to the COVID situation, will be through either commenting this video on YouTube or putting your comments on the Neurostar forum, like I indicated in the pamphlet of the course. And a little project that I'm going to ask you to do, which is writing an essay between 1,500 and 2,500 words, where you gotta have to choose brain hierarchy you want to write about, and write your essay on this brain hierarchy in 5 different sections, including a general introduction where you gotta get me excited about it. Why is it interesting? Please convince me. The anatomy of this hierarchy, whether this hierarchy is lateralized, and if it's not reported that it is lateralized, how would you investigate its lateralization really? And fourth section, which will be about the link between this hierarchy and brain evolution. And finally, I'd like you to give me a full synthesis of the previous part. Show me how anatomy, lateralization, and brain evolution in this hierarchy work together. Again, this is 1,500 to 2,500 words in total, not including the reference of the manuscript. And the deadline will be December 11th, and it will be, and you will have to send it to me at my address, michel.thiebaud@gmail.com. Thank you very much for your attention, and I look forward to teach you the next course on the holism.